Hi student, Assalamu alaikum. Today our topic is on structure of the ovum and cleavage of the zygote. Ovum is 1000 times larger than the sperm, and this stage of the ovum is chondriocyte stage. Ovum consists of three most important membrane. Innermost membrane is Vitellian membrane, which consists on phospholipid, and second membrane is zona pellucida, is thick and most important membrane, which is made up of the glycoprotein, and corona radiata is the third and outermost membrane consistent follicular cell. The function of these three membrane is that Vitellian membrane only allow one sperm. For fertilization only allow one sperm, and zona pellucida is prevent the unwanted sticking of the fetus or zygote. If these zona pellucida rupture in the fallopian tube before the implantation in the uterus, due to the rupturing or breaking of the zona pellucida, these zygote attach with the fallopian tube of the mother. That is a result in death of the mother take place. So corona radiata consists of follicular cell. Follicular cell is attached with each other with the help of the hyaluronic acid. Hyaluronic acid is dissolved through the hyaluronidase enzyme. These enzymes is released through the acrosome of the sperm. During fertilization, sperm release that enzyme. These hyaluronidase enzyme dissolve the hyaluronic acid, and sperm reach to the zona pellucida. Due to the dissolvedness of these hyaluronic acid, and follicular cell separate from each other and take path for the sperm. So corona radiata release corona lysine enzyme that break the follicular cell, and zona pellucida release the zona lysine enzyme that dissolve the zona pellucid membrane. So due to dissolvedness of this membrane, sperm enter into the Vitellian membrane. Vitellian membrane consists on the cortical granules. This bead-like structure is called cortical granule. These granule allow formation of the reception cone. This is the reception cone that receive the sperm. After receiveness of the sperm, Vitellian membrane close itself, and not allow another sperm to enter into the ovum. Reception cone is rotate at 180 degrees centigrade that help the sperm enter into the pronuclei of the ovum. After entrance of the sperm into the pronuclei, fertilization take place. Now after fertilization, zona pellucida become more thick and thick. So now ovum called zygote. Now ovum called zygote. Single cell ovum now called zygote after fertilization. Now cleavage or division of the zygote start. Compaction of the zygote start. Cleavage is the division in which number of the cell is increased but the size of the cell is remain same, in which number of the cell is increased but the size of the cell or zygote remains same. In this type of the division, in which after 30 hours zygote divided into two cell. In this type of the division is meridional plane. In meridional plane division is called vertical division, and after 60 hour equatorial plane division is take place. 4 cell stage is formed, and 8 to 60 cell stage formed after 3 to 4 days. This cell stage is called marula. Now zygote become marula after 3 to 4 days after the fertilization. Consists on 8 to 16 cell. Marula is mulberry like structure. After marula, blastula is formed. Blastula formed like that. Smaller cell goes toward the peripheral side of the fetus, go to the peripheral side of the zygote, and larger cell go to the center of the zygote. This type of the division is called blastula. Blastula consists on 32 cells stage, and after blastula, blastocyst is formed. Blastocyst is in this blastocyst, zona pellucida start break and dissolve during blastocyst. Zona pellucida break for the implantations of this zygote. Smaller cell, peripheral smaller cell is called trophodrome, that made the placenta, and that made the placenta of the mother. And larger cell, innermost bigger cell is called embryonic knob, that made the embryo or three germ layer: ectoderm, mesoderm, endoderm. In future, that make complete fetus, complete baby. In future, this embryonic knob, innermost cell, made three germ layer. One baby or organism, living organisms consist on three germ layer: ectoderm, mesoderm, and endoderm. These embryonic knob made in future three germ layer. So this is the cavity is formed, blastocele cavity, that filled with the uterine fluid. Uterine fluid is present in this cavity. This fluid consists on the food that give to the embryonic cell. Embryonic cell feed this uterine food through the blastocele cavity and grow itself and divide itself continuously until the attachment, until the implantation to the uterus of the mother. So this is the complete lecture on the cleavage and the structures of the ovum. Hope you understand it. Okay, Allah Hafiz. Like and share.